Hi, hello everyone. My name is Emiliana Weiss, and today I'm going to present my research project, which the title is Variability and Transcriptional Properties of Human rDNA Repeats using long-read sequence technologies. So my personal focus in my PhD is to investigate and elucidate the variability of ribosomal DNA. And ribosomes, they are essential, as we know, for protein synthesis in all living organisms, and most of the genes that encode for this machinery are located in a tandem repeat array in 5 human acrocentric chromosomes. And despite the completion of the human reference genome, the rDNA repeats are one of the last remaining gaps in the human genome and have so far resist complete assembly. Currently, rDNA studies, they use what we call consensus rDNA repeats and assume that they are invariant. So in here, there are the 2 gene bank annotations code. And the resistance to assembly is caused by the repetitive nature of rDNA and the type of technology being used to sequence the human genome. So for a long time, we have been using what's called short sequence reads. So the short sequencing, short sequence, they produce reads with 250 kb, around 250 kb, and it's very difficult to trace back if a read belongs to one rDNA region or another. And to overcome this situation, this limitation, third-generation sequence technologies such as Oxford nanopore have been proposed, and to do by using these ultra-long reads that are produced sometimes with greater than 100 kb, we can transverse complex and repetitive regions such as the rDNA arrays. So in here, the main point of this project was to develop a methodology to characterize ribosomal DNA, which is essential to determine the impact of rDNA variants on cell phenotype. And to do that, we have developed a pipeline. You can focus in here if you want to know a little bit more. And the dataset that we are using belongs to the Human Pangenome Reference Consortium, the HG002. And basically, the pipeline consists of mapping this FASTQ reads, the HG002 reads FASTQ files to the human genome. This human genome is a modified version where a tandem copy of the human rDNA repeats were introduced as an extra chromosome, and all the regions that are rDNA-like in the human genome were hard masked. So in here, I'm just plotting that there are a lot of regions in the human genome that are rDNA-like. Then all the reads that were rDNA were selected, and a further investigation about methylation was done by using the tool Methylation, and also annotation using BLAST to observe a structural rDNA variation. So in here, what I'm showing is that we're one ultra-long read, so we were able to recover rDNA units in noise ultra-long nanopore reads. So we were able to identify 918 reads with length greater than 100 kb, containing a total of 3,300 rDNA candidates. In this picture, what I'm showing is transcribed rDNA regions across the rDNA reads that were filtered. So in here we have 7 rDNA units. And by analyzing all the reads filter, we could observe that rDNA units had a highly conserved site, suggesting that most rDNA units have full coding potentially. So I'm showing this in this graphic. And to do this analysis, what we have used was the distance between two regions in the rDNA units. Moreover, now let's change and talk about the methylation investigation. One thing that's very remarkable that we need to remember is that in eukaryotic cells, only half of the RNA gene copies account for transcription of cellular RNA. And because of this, we decided to further investigate it. And we have analyzed the raw signals of nanopore sequencing to identify DNA CPG methylation. We have used the tool Methylation to predict the pattern of CPG methylation on those reads, and we were able to distinguish 2 contrasting patterns with similar proportion, around 50% each one. So in this first pattern that I'm showing here, what you can observe is that the promoter region were methylated, and the IGS is highly methylated, and this may indicate that transcriptional active genes. So in here, all the genes would be transcribed of these rDNA units. And in the second part, and what we have observed is that the RNA genes, the promoter, the IGS, were highly methylated. So we have a highly methylated signal, indicating that these RDNA units that belong to this signal, they are possibly not transcriptional. So there is a transcription repression. A repression transcription. And moreover, we have found that reads with there are some reads with inversion. So there are some units that are transcribed in this direction, and two other units that are transcribed in this other direction. And what we have observed in that methylation is that we have a repression in these two units, what may indicate that they are possibly not inactive. We also have investigated presence of sequence variants in the rDNA. And to do that, we have used PacBio Hi-Fi reads from the same data set that's available on the Human Genome Reference Consortium. And by using the same pipeline, we're able to identify a total of 2,925 variants that are distributed in, and I'm showing this plot differently in the rDNA regions. After this analysis, we have decided to investigate the rDNA regions, and we have then performed our rRNA sequence using mini-ion from the cytosolic fraction of the same cell line. And we found that 99 of the rDNA variants were also expressed in cytoplasmatic rRNA. And furthermore, many variants present at a low frequency in the rDNA were also found in the rRNA with high relative abundance. So finally, we can summarize with some of the key finds. So first, it was possible we have successfully filtered rDNA reads from the whole human genome dataset. Secondly, we have identified an exceptional level of variance across rDNA repeats, including sequence substitution that are expressed in the variant RNA molecules. We have found structural rearrangements that affect orientation of RNA genes and non-randomic organization involving two contrastly patterns of DNA methylation, and where some units in the same region can be repressed or also active. So I would like to thank you, everyone, for being to listen to this presentation, and all my dear friends from John Curtin School of Medical Research.